Okay, so we are continuing in this fourth video regarding part 1, chapter 2, from slide 44 of the presentation. So when proteins of different organisms have the same structure and function um, due to an ancestor origin, it is said to be a superfamily of genes. Okay, so superfamilies exist. They're usually very big and very wide and have branches and different species and can be tracked back to that ancestor origin, or in our case, ancestor gene. So those kind of genes are either identical or extremely similar in sequence in their map and in their structure. If the superfamily evolves to add functions, a common structure can still be identified. So it doesn't mean that they are identical. The superfamily can evolve and produce different functions in different versions of variants of the genes, but you'll always be able to identify a very specific structure that is the same for everyone. So, for instance, on slide 45, we are taking a look at the globin superfamily, which is an interesting superfamily. So we see that we have hemoglobin, we see that we have myoglobin, and we have what we call leg hemoglobin. And underneath that, I've just given you kind of a computer module of the structure, the 3D structure of the molecule, and you can see that they're not exactly the same. But within each and every one of them, if you start analyzing them, you'll find a common structure. Okay? So for instance, myoglobin is about 1/4 the structure of hemoglobin. So you can actually say that hemoglobin is more or less 4 times the myoglobin combined together, and so on and so forth. So we are on slide 46. So that common feature helps us identify the superfamilies and also the ancestral gene or origin, and also understand what were the phases that structure that the family evolved through. When we find this extensive discrepancies between the superfamily, that means that that gene must have underwent extensive removals or insertions, right, of introns, because you'd expect theoretically for a superfamily to evolve in a similar way. Of course, that can be related to the needs of the organism, where it's living, and so on and so forth. All right, so we are on slide number 47. And the idea is that at present, a single axon can be the result of a fusion between 2 separate axons in the past. So imagine we had 2 axons, and I'm just making this up to make it easy to understand, of 100 base pairs in sequence. Okay? And for some reason, they are connected through evolution. It happens, and those 2 axons, 1 and 2, are connected. Now suddenly we have an exon that is the combination of both, so it's 200 base pairs, right? In the example only. And we get a new ability because the meaning of the fusion changes the function, right? So it's no longer read as 2 exons rather than 1. It is possible that that axon has simply been inserted during evolution. It's always a possibility. So we can either have fusion or insertion, right? And it is sometimes assumed that the ancestral gene had more introns than the present genes. So the assumption is that most likely that during evolution, introns are taken out. So if we try and imagine an ancestor gene being the tree and the present genes being the branches, so you have that stem, which would be the ancestor gene, and anything that you find afterwards will be the branches, and the branches don't have to be the same, right? But they're still part of the tree and they're still originating from the same stem. Alright, so one can actually imagine that maybe the ancestor gene did not have any introns at all. So we're playing with theory and possibilities. One is that it had more introns than the genes today, and the other possibility is the other way around. It had no introns, and introns were actually introduced during evolution's cycles. So which one is the truth? We don't know. Otherwise, it would be defined, right? It is also possible that the number of genes that existed in the past is way lower than the number of genes that exist right now. Because we're talking about duplication, we're talking about variation, right? We're talking about recombination. Those are mechanisms that may produce variety. So it's possible that with time, those mechanisms, those procedures, those phenomena took one gene and evolved into 5. So perhaps in the past, proteins did not have as much modules and functions as they have now. So creatures would be less complicated in a way, right? So we are on slide 49, and the major thing written here, and I want to emphasize that because it has great importance to why this course is taught to you guys and why this course is important to anyone that wants to be a professional, is that we have advanced our knowledge and understanding now that the use of the idea, the hypothesis of one gene, one protein is incorrect, right? We can definitely understand that. So it is clear that the gene is subjected to further processes, processes that might be exact or mistakes, right? That can lead to various outcomes and expressions. That's a major title or understanding that you must have by now. Okay, so I hope you do. So when we studied last year CP 636 and 637, clinical exercise physiology, advanced clinical exercise physiology 1 and 2, we noticed that most of the chapters and the episodes and the issues, the topics, were giving great detail to cascades. So we understand that gene expression is a cascade of different processes and reactions that are supposed to be executed, done in a certain order and to a certain extent of accuracy, right? So the second word that we must understand is a cascade. And that relates to the question that I asked you in the first presentation, trying to explain to you that if you can't answer my question within a few seconds, you need this course. We are understanding the complexity of reactions, and we understand that gene expression has a lot to do with adaptations. And when we are trainers, what are we doing? We are applying some kind of manipulation, so to speak, right? And expecting a certain adaptation. And in between, we have cascades that are happening, influencing gene expression, thus seeing the outcome. So this course is a very important link between the manipulation and the outcome, the adaptation, right? So gene expression or inexpression, right? A gene can be expressed and it can also not be expressed, is the result of a cascade of reaction and processes in the cell. Does that make sense? I hope it does. So from the beginning of creation of a new organism, gene expression cascades occur and are timed accurately due to regulation mechanism. Meaning that when we take a human baby or a human fetus, are all the genes that are expressed as a fetus also expressed as an adult or an advanced age adult or an adolescent? No, of course not. That means that we have mechanisms of expression that are timed, and they must be timed accurately because some of them have to be aligned perfectly with our development. So that's very interesting if you ask me. How does the mechanism know the exact time when to express a gene? Just imagine what accuracy we would need. Okay, so those mechanisms are greatly still misunderstood or even unknown. I think that once that is broken down and understood, magical things will happen, because I mean, just think about it. After what we've learned in only 3 chapters, or in 3 presentations, we understand what great mechanisms, what complex mechanisms, and how important accuracy is. So, of course, the genes that allow certain abilities must be produced and expressed in the correct order. Otherwise, development as we know it will be completely different, and most likely for the worse, right? So it is now clear to us that our genome includes information for regulation mechanisms of gene expression as well. It's not just the proteins. We have to understand the proteins are important, right? But also regulation and timing are important. So we're talking about a gene that includes information for function, information for timing, information for regulation. Okay, that's very, very important. And it's extremely safe to say that genes actually interact with each other as part of their expression. So the expression of one gene may be the regulation mechanism for the expression of another gene and its function. Okay. So we can talk about positional information. Positional information relates to the knowledge that certain factors regarding regulation and development are located at certain locations of the cell. So the idea of location of different processes in the cell is not new. We know there is divisions as a principle in the cell, right? We have the cytoplasm, we have the mitochondria. They're conducting different processes, and the same goes for what we call positional information. So different areas conduct different processes. And by the way, we've seen that in regards to gene expression, because if you recall, transcription and translation are not done in the same place, right? We talked about the mRNA needing to leave the nucleus and go to the cytoplasm for it to happen. So it's also possible that part of a gene regulates or determines where the information is going to be located. More than possible. So that would be part of regulation. Okay, so the gene may include instructions of where the information is extracted and used, which is very important. So when we're talking about the development of an organism, we may actually define it in such a way. The accurate and controlled gene expression of the proper genes. Okay, so with that being an important point, we will finish this video and chapter, and I'll see you again.